Today, we're going to explore a fascinating and powerful class of natural compounds, alkali. At their core, alkalides are simply organic compounds that are found in nature. What makes them unique is the presence of at least one nitrogen atom, which is usually integrated into a ring-like structure. While we most commonly associate them with plants, they are also found in fungi, bacteria, and even some animals. The term alkaloid itself dates back to the early 19th century and comes from the Arabic word alkali, which means ashes of plants. This name was first used to describe these plant-derived substances that had a basic or alkaline nature and significant medicinal effect. From a chemical perspective, alkaloids have a defining property, their basicity. This comes from the nitrogen atom, which acts as a proton acceptor. This is a crucial characteristic as it allows them to react with acids to form salts. Their basicity directly impacts their solubility. In their free base form, they are highly soluble in organic solvents like ethanol or chloroform, but not very soluble in water. However, when they form salts with acid, their water solubility increases dramatically. This unique amphiphilic characteristic is absolutely essential for the extraction from plants and for formulating them into various medicinal applications. Beyond their chemical makeup, alkaloids play a vital ecological role in the natural habitats. For plants, they're powerful defense mechanism. Think of the bitter taste of quinine or the toxicity of nicotine. These are nature's own chemical deterrent against animals and insects that might try to eat them. For example, morphine in the opium poppy helps protect the plant from microbial attack, while nicotine in the tobacco plants effectively discourages insects from feeding. Alkaloids are classified in a few ways, but the most common method uses their chemical structure and the specific amino acid they are derived from. We can see some of the major classes right here. Indole alkaloids like vincristine come from tryptophan. Isoquinoline alkaloids such as morphine are derived from tyrosine. Tropane alkaloids including atropine originate from ornithine, and then we have classes like purine alkaloids, which includes caffeine, and quinoline alkaloids like quinine, each possessing a unique structural backbone. Each of these distinct structural classes also boasts a specific set of therapeutic application, which naturally leads us to explore their various sources and uses. Let's dive into where we discover these compounds in nature. Plants are the most abundant source. It's estimated that over a quarter of all flowering plant species produce alkaloid. Some of the most renowned examples come directly from plants we've long utilized in medicine. The opium poppy, for instance, provides us with morphine and codeine, two of our most powerful pain reliever. The bark of the cinchona tree is the original source of quinine, a vital anti-malarial drug that has saved countless lives. And from the Madagascar periwinkle, we get vincristine and vinblastine, two cornerstone chemotherapy drugs crucial in cancer treatment. But plants aren't the only kingdom contributing to this rich family of compounds. Fungi are well-known producers, such as the ergot alkaloids from the Claviceps purpurea fungus, historically used to treat migraine. We also find them in certain bacteria and, surprisingly, even in a few animal species. For example, marine conch snails, which produce potent neurotoxin conotoxins, currently being researched for their potential in pain management. Let's focus to how these remarkable molecules are created. The process, known as biosynthesis, is a complex journey with many steps that happens right inside the organism. It generally kicks off with simple amino acids acting as the initial building blocks. Different amino acids serve as starting points for different types of alkaloids. For instance, tryptophan is the foundation for indole alkaloids, while tyrosine leads to isoquinoline alkaloids. These amino acids then go through a series of chemical changes like decarboxylation and condensation to gradually form the intricate structures we observe. This origin in biosynthesis also gives us another way to classify alkaloids. True alkaloids, such as morphine, come directly from amino acids and have nitrogen nestled within a ring structure. Then, there are protoalkaloids, like mescaline, which also start from amino acids but don't have that nitrogen in a ring. Finally, pseudoalkaloids such as caffeine do contain nitrogen in a ring, but they aren't built from amino acids. Understanding this detailed pathway is incredibly important for researchers. Perhaps the most exciting aspect of alkalides is their wide range of biological activities. Their unique structures allow them to interact with a variety of molecular targets within our body. We can group the therapeutic effects into several key categories. For instance, in the realm of antimicrobial actions, compounds like berberine are potent against bacteria, fungi, and viruses by damaging their cell membranes, a truly promising area in our ongoing fight against antibiotic resistance. In the fight against cancer, vincristine and vinblastine stands out as fantastic example. Their work by preventing cancer cells from dividing, a mechanism that has saved countless lives through chemotherapy. As for pain relief, morphine is the classic example. It's one of the most powerful analgesics in medicine, working by targeting opioid receptors to modulate our perception of pain. Compounds like palmitin help protect our cells from oxidative damage, a crucial factor in preventing chronic diseases. Finally, galantamine is used to treat Alzheimer's disease by influencing neurotransmitter, and we're all familiar with the stimulating effects of caffeine and even nicotine on our cognitive performances.
Alkaloids are among the most impactful natural products in modern therapeutics. Their nitrogen-containing structure gives rise to a wide range of pharmacological activities. Historically and currently, they are used directly as medicine or modified to develop semi-synthetic drugs. Their therapeutic roles span from pain management and cancer treatment to infectious and neurological disease. One of the earliest and most effective alkaloid medication is morphine. Extracted from Papaver somniferum, it is a potent opioid receptor agonist, providing relief from severe pain. Codeine, also derived from the same plant, is used for mild to moderate pain and as a cough suppressant. To improve bioavailability and reduce side effects, semi-synthetic derivatives like oxycodone and hydromorphone have been developed and widely used in clinical settings. Alkaloids also play a vital role in oncology. Vincristine and vinblastine, indole alkaloids from Catranthus roseus, inhibit mitosis by binding to tubulin, making them effective in leukemia and lymphoma treatment. Another major anti-cancer alkaloid, camptothecin, interfere with DNA replication by inhibiting topoisomerase. In addition, paclitaxel, to a diterpenoid alkaloid, stabilizes a microtubule and is extensively used against solid tumors such as breast and lung cancer. Infectious diseases have also been treated with alkaloids. Quinine, isolated from Cinchona officinalis, was a historic breakthrough in malaria therapy. It became the prototype of synthetic derivative like chloroquine, though not an alkaloid by strict definition. Artemisinin shares similar application and is central in modern antimalarial protocol. This example underscores the pivotal role of alkaloid in global health. To ensure the safety, efficacy, and consistency of alkaloid-based drugs, reliable analytical techniques are essential. These include procedure for extraction, purification, structural elucidation, and quantification. The complexity and variety of alkaloid structure demands sophisticated multi-step workflow. Traditional extraction of alkaloids involve acid-base partitioning. The alkaloids are first converted into water-soluble salt in acidic condition, then re-extracted as free bases using organic solvent like chloroform. Purification techniques such as column chromatography and recrystallization help isolate specific alkaloid from plant mixture. High-performance liquid chromatography, or HPLC, is the most widely used method to quantify alkaloid in raw extract and drug formulation. It provides high resolution, separation, and reproducibility. For volatile alkaloid, gas chromatography mass spectrometry or GCMS is used. It helps identify compounds by analyzing their mass spectra and fragmentation patterns, especially in forensic or pharmacokinetic studies. Nuclear magnetic resonance spectroscopy or NMR is the gold standard for determining the molecular structure of alkaloid. It reveals how atoms are arranged and connected, including the stereochemistry and functional group. Techniques like 2D NMR, COSY, HSQC, and HMBC enable in-depth analysis of even complex structure like vincristine or morphine. Besides classical technique, newer methods are gaining popularity. Capillary electrophoresis (CE) provides high-resolution separation with small sample volumes. UV VIS spectrometry is quick and cost-effective for initial screening. Biosensor and ELISA-based immunoassay offer real-time detection of specific alkaloids like nicotine and morphine, even in clinical and environmental sample. Recent research is focused on discovering novel alkaloids from underexplored sources like marine organisms, fungi, and lesser-known plants. These new compounds have shown anti-cancer, anti-inflammatory, and neuroprotective activities. Structural modification of existing alkaloids, such as competition derivative, have improved water solubility and reduced toxicity, demonstrating the importance of chemical innovation. To meet rising demand and reduce ecological pressure, biotechnological methods are now used for alkaloid production. Plant cell culture, hairy root system, and engineered microbes like E. coli and yeast can produce compounds like scopolamine and morphine sustainably. This approach provides scalable, environmental-friendly alternative to harvesting wild plants. Despite these advances, several issues persist. Over-harvesting rare plants for alkaloid threatens biodiversity. Resistance is growing to antimicrobial and anti-malarial alkaloids. Future strategies must focus on designing drugs with novel mechanisms and higher target specificity to reduce off-target effects. Sustainable sourcing combined with synthetic biology will be essential to overcome these challenges. To conclude, alkaloids remain indispensable in drug discovery and therapy. Their structural diversity and strong biological activity make them valuable tools across medical fields, especially in cancer, pain, infectious disease, and neurology. The future of alkaloid research lies in the integration of computational drug design, synthetic biology, and advanced analytics, which will enable safer, more effective, and sustainable drug development.